Hello, everyone, and welcome to the latest and greatest episode of the Richard Watkins podcast. I am your host, Richard Watkins. Before we get started, we have to give a huge shout out to the folks that make this possible, the Science Policy Action Network. The world is changing all around us very, very quickly, and you might have questions or might want help with accessing opportunities, regardless of whether you're in government, private sector, academia. It doesn't matter. It has anything to do with education, health, for innovation. SPAN is here to support you. Find out more at span-inc.org. That's span-inc.org. Without further ado, let's get the proceedings proceeding. This is absolutely an honor. We have a great guest. We have Dr. Vijay Sivaraman. Today's guest is Dr. Vijay Sivaraman, an associate professor in the Department of Biological and Biomedical Sciences at North Carolina Central University. He is also a researcher affiliated with the Biomanufacturing Research Institute and Technology Enterprise, better known as BRIGHT, and the Julius L. Chambers Biomedical and Biotechnology Research Institute. Dr. Sivaraman research explores how environmental exposure and substance use, like alcohol and cannabis, impact lung health and immune response. His work sits at the intersection of pulmonary biology, immunology, and public health, with a deep commitment to understanding how lifestyle and environment shape respiratory diseases. We're thrilled to have you. Hopefully, I got that bio right. What did I miss out? What should I have
Hey, you
changed?
made me sound a lot better than I think I am, but that's pretty good. That's pretty good. I'll take it.
That was good reading on my part. If you saw me looking off screen, I was like, let me get this absolutely right. Hey, listen, it's an honor and treat. For the folks who are listening or watching, I got a chance to get to know this man a long, long, long time ago. I know we both look pretty young, but we've been in the game for a long time, listening to him do presentations at the University of North Carolina at Chapel Hill regarding the pneumonic plague. And now he's at North Carolina Central University. Tell us what you were up to.
Well, what am I up to? Right now, I am evaluating airway samples and how human airway samples and how they were exposed to a variety of environmental contaminants. So, in today, I was running ROS assays on cells that were exposed, human bronchoepithelial cells that had been exposed to um, microplastics, specifically tire leachate, which is a major project that I'm interested in pursuing right now. And I'm doing this in collaboration with Research Triangle International. And so we're looking at tire leachate as one of the major microplastics that we're exposed to via an aerosol exposure. If you think about it, you're walking down the street on a sidewalk, you're not thinking about the fact that the air you breathe is containing microparticles that are coming from the tire debris that's grinding off those tires from those big vehicles driving by you at unnecessarily fast speeds. And that's sloughing up into the air. It's ground into very small particle sizes of a particularly between PM10 or 10 nanometers and 1 nanometer. And when that goes into your airways, when you're just taking a breath of air, it goes deep into your alveoli. And it doesn't break down. The problem with plastics are that there's nothing organic about them and they reside in your alveoli, and where it's leading to unknown damage, unknown distress. And so we are evaluating on an acute level and ultimately a chronic level what happens when we get these particulates in our airways. So we can do that acutely with human airway cells, which I'm working on right now. And also, we can do long-term studies with murine models because we can actually vaporize this tire particles and expose them intranasally to mice. So we can evaluate that over chronic exposure, how it leads to pulmonary dysfunction, distress, potentially COPD.
We had on the show Dr. Viswet at UNC, and he was talking about particulates and roughly the same thing, their impact on health. But he also talked about how, but in layman's terms, air quality is worse when you live in areas that are impoverished. Does your work touch on that at all? And if it doesn't, please forgive me. I'm just curious.
Very much so. As you can see from my backdrop, I'm at NCCU, North Carolina Central University, and our major focus as an HBCU, as the premier HBCU, I'll say, for research in biomedical science. Okay, that's great, and you're a great institution, but you're not the premier biomedical research institute. When Fayetteville State is situated in RTP next to UNC and Duke University and NC State, we have a little bit more credential, right? So, okay, so what are we doing with the Bright and BBRI institutes? We have the capability to ask some questions that have to do with health disparity. That's our major focus. The research motivation behind NCCU is primarily focused around diseases of health disparity and how we can intervene. What kind of interventions can we put in place to better our community? And so I am a member of our RCMI funded center to focus on health disparity research. And so this is a question of health disparity. We recently published a paper in Frontiers of Public Health, specifically evaluating the airway exposure, particularly asthma exacerbation within North Carolina. So I was able to acquire hospital admissions and discharge data from of the North Carolina hospital systems for the span of 5 years, over 2012 to 2017, pre-COVID. And we were evaluating asthma exacerbation within these populations at a zip code level. And we found that the zip codes around Durham, North Carolina had the highest level of asthma exacerbation across the state of North Carolina. So then we dialed it in to figure out which zip codes within the 12 zip code region had the worst. It was 27701, which happens to be a budding NCCU. So then we took it to a granular level because zip code is a big space. So we actually broke it down to census tracts. And we found that there were census tracts that which we call South Durham, which were the highest, 97% African-American constitution, and also the northern census tract, which we use, which was predominantly Caucasian, the lowest incidence of minority. And when we look at the hospital admissions data within that zip code, we found that the highest incidence of asthma exacerbation was with the African-American community. So we then wanted to ask the question of what was going on within these regions at the granular level of the census tract, because the EPA reports over a wide region what our PM level of exposure is. So when we get that data, we get from AirNow and whatnot, other resources, we know from the level of several zip codes overlaying, not so much the granular level of neighborhood to neighborhood. So we proposed to evaluate on that granular level. So we made community partnerships within the northern and the southern census tracts. We actually set up air collection monitors outside in people's homes, or outside businesses really, and churches. And then we quantified and we collected the air particulate, specifically looking at particulate matter 2.5 size nanometers, which reside, as I said earlier, into your alveoli. So that project led to this paper because we found that on a granular level, there was a higher level of PM 2.5 within the minority in communities, suggesting that there's road use, development, elements that surround redlining that leads to worse air quality within minority neighborhoods, lower SES communities than the PWI institutions and environments. And so there was the element of quantity, but there's also an issue of quality. We wanted to evaluate the actual particulate matter and see if there was a difference there. And so we were able to take the collected particulate matter from these regions and then run them through a array of analysis. So we evaluated them for inorganic materials. We were able to run transmission electron microscopy on these samples and found there was a difference in inorganic materials across these sample sets. We were also able to evaluate microplastics because that's in the air as well. And so, as I was saying earlier, and that's kind of what opened this whole pathway, this whole can of worms of studying tire leachate, because now we are finding that there's differences in the microplastic constitution and the potential points of origin, neighborhood to neighborhood, that contributes to this asthma exacerbation within our community. So
that is absolutely fascinating. As a quick question, so when you're taking air samples and you're saying you're taking air samples from around North Carolina Central, and I'm imagining you're taking samples from around Duke University, and you're looking at both quantity of particulates and the composition. And if that understanding is correct, um, is there a higher concentration of just particulates in general in areas closer to North Carolina Central, or is it just, say, for instance, the bad particulates, or all particulates bad? I guess we should back up. Just back up. Are all particulates bad? I think I should ask that.
So I can't answer that question of whether there is a spectrum. I can say that some particulate matter is worse than others, depending on the constitution within that particulate matter. So level is important, but constitution is also important. So we have exposed these particulate matters collected upon airway cells, cells that are, derived from upper and lower airways. And we found that there is somewhat of a differential pathology associated with these collected air samples. So that's what has led us to evaluate what is actually in these air samples to do these exposures.
God, that is fascinating. I mean, I've always loved your work, not only your work, this is my opportunity to tell you, I've also loved your presentations, going all the way back to graduate school. I actually want to touch on that really quick because a lot of people, they hear about research, they hear about these studies being done, but I think also important is understanding the person doing this type of research. And you've been doing this type of research, understanding, respiratory systems and the impact that environment or just different, I don't know, damages to them for quite some time now. I mentioned at the very beginning in graduate school, you did work on pneumonic plague, right? Is that understanding correctly? And then how did that understanding inform the work that you do now, and how does a researcher, and this is for any young researcher, how do they then make the pivot from maybe their graduate and postdoctoral work to now the work that you're doing now as an associate professor? How does that even happen?
Well, that's a good question. I mean, it's an interesting path or storyline. I would say I've been stumbling down the road of academia for quite a while, but my PhD work was really focused on. Okay, let me back up. So post undergraduate, because I went to a hippie college, and I fell in love with the idea of gene therapy and working with creating viral vectors to express proteins, all that, while I was an undergrad. I actually did a summer undergraduate research experience at UNC in the gene therapy group that taught me that. So then I was excited about science. And so I pursued a post baccalaureate fellowship with the National Institutes of Health. And I worked in the context of the infectious disease center as well as the eye institute. So I worked in a collaboration between NIAID and NEI, and we developed understandings of autoimmunity within the eye. And it was an interesting, fantastic 2 years of my life where I got to go to exceptional talks and meet exceptional scientists and learn immunology. And that led me to graduate school at UNC Chapel Hill. And while I was there, I fell in love with HIV pathogenesis because it's a virus that seems particularly opine to attack the immune system. So it's an interesting battle between pathogen and host. So that was an incredible time where we did a lot of good work and got some great stories out of it. And I have to say, I'm proud that I was involved in some of the early PrEP studies. So it was pretty cool stuff. But that made me realize also that I didn't necessarily want to work in HIV for the rest of my life. And I didn't want to work with something that could kill me necessarily. So I went into my postdoctoral fellowship realizing I cared about immunology and specifically in the airway. And I wanted to move into that direction. So I moved into a lab that studied non-pathogenic things that cause immune activation in the lungs, but really got swayed by the more sexy side, which was the Yersinia pestis side. Why? Because it causes a fulminant pneumonia that kills you in 72 hours. So it was exciting. I did that work. It was really great. But the same question came up of like, do I want to do this for the rest of my life? And I didn't. I wanted to study the understanding. I wanted to develop an understanding of the immune response in the airways with the ability to target and evaluate and potentially come up with therapeutics to impede that. NCCU gave me an opportunity to be an immunology professor here and also create a research program where I could involve minority students. And I was very excited about that opportunity. So while I came here, what the question that came up constantly was that we all get microbial infections, right? But some get disease worse than others. And so that led us to understanding what environmental and social factors contribute to worsened disease outcomes when you get that strep pneumonia, when you get that influenza, when do you get that coronavirus? You know, all those things happen to everybody for the most part, but some have worse disease than others. So that has led us to evaluate alcohol intoxication, binge drinking. Some college students, you didn't do this back then, I'm sure, but some college kids, they drank too
Oh, no,
much.
no, no, no.
No,
Let's
no,
set
not
the
you.
record straight. Yes, I did. I was a regular person before I became a dad and a scientist. So this is important for everybody. So right back at you. I'm sorry, I'm not perfect like most people think I might be.
Okay, okay, okay. Uh, but I'm just saying that, you know, for those who party for the weekend in college, there's a high incidence of pulmonary distress. Okay, how about I put it this way? Alcoholics present to the clinic with pneumonia more than any other reason. So we wanted to evaluate why pneumonia was the distress for those who have a high alcohol incidence. And so that had led into the question of alcohol intoxication, and as a mediator for increased inflammation, specifically when you get an infectious disease.
I just asked that because you just dropped something there that was you said, and I don't want to say it wrong. You said alcoholics present in the clinic with pneumonia more than any other health complication. Did I
Correct.
understand you correctly?
That is correct. That is correct. And so, because of that clinical finding, that led us to ask this question: what happens? Can we develop an animal model to evaluate college age binge drinking and how it leads to downstream disease burdens? So, we did the basic question of taking a mouse, giving it oral gavage of alcohol, you know, day on, day off for a span of 10 days. It's a standard NIAAA model of acute intermittent ethanol exposure. So we did that, and then we let them rest for 12 days, which is kind of in a mouse world, like moving into like early adulthood, like in your 20s, 30s. And then we challenged the animals with a sublethal dose of a Klebsiella species, which in an unexposed animal or a water exposed animal gives them a mild inflammation. They recover and they're fine. Much to our surprise, the animals that had been primed with ethanol 12 days prior, 12 days prior, they had severe pneumonia and many of them died. Okay, we had a high mortality rate. So it was a crazy model that allowed us to parse this out. So this story has led us down a rabbit hole of molecular understandings, and we have found that somehow cannabinoid signaling plays a role. Cannabinoid signaling is the way cannabis also activates cells within our body, brain, lung, liver, to signal for activation for responses. So the way cannabis works, potentially alcohol does something similar, but not similar. We're trying to figure this out. So this is actually an interesting story, a major NIH-funded project that we've had worked on for quite a while. Keep an eye open for Frontiers in Immunology. We're going to have a paper out soon. It's pretty cool. And so, so that's one major arm of my research program. Another major arm is e-cigarettes, because e-cigarettes came on the scene about 10 years ago, and really there was no regulation. So we've been evaluating that to understand and better inform FDA and people who listen of elements of toxicity within e-cigarettes. You know, we've been focusing on Juul menthol and other menthols because that is unfortunately the type of tobacco that is pursued by minorities at the highest incidence, and we find that it's also the most pathologic. So we're trying to discriminate why that is the case so we can better inform our communities and come up with potential therapeutic interventions. So that's a second arm. And then the third arm is the air quality project that I told you about earlier.
This is all just super incredible. All right. So the first one, you're looking at cannabis use, you're looking at alcohol consumption and its impact on the likelihood that somebody will develop pneumonia. Now, I kind of want to go back and ask something about that situation with pneumonia. Is it upon like infection? And do you see a situation where individuals have increased likelihood of super infection because of the damage already done to the lungs? Or does that not have anything to do with it? I'm thinking like some of the things that we might see with RSV, where you might have a super infection because an individual is not able to properly clear mucus, from the lungs. Do we see the same thing there? And it's been a long time since I was in microbiology. So if I'm getting anything incorrect, please don't hesitate to correct me. But is that any of the stuff that you see?
Okay, so what do we see? We see in the case of alcohol, of animals that have been priorly exposed to ethanol, the microbial replication is a good log to 2 logs higher. So there's more microbes in the airway. Okay. There is not an increase in mucus per se, but there is an increase in just filament inflammation and neutrophilia that comes in the airways. So the animals, when they've been imbibed with ethanol, and cannabinoids for that matter, don't necessarily get sick by themselves. But if you give them an illness, that illness is far more severe in manifestation. And that suggests to us that you have a direct inflammatory effect that is being primed by the prior exposure and exacerbated by infection.
That is brilliant. Troubling, right? Troubling because it shows some of the things that we take for granted, right? Some of the things that are traditional pastimes, activities of leisure, they could down the road impact how you recover from an infection. I mean, that's just absolutely incredible. Now, when you are seeing a situation where somebody is dealing with an infection, is there one infection that is more problematic than another? Say, for instance, RSV is more problematic than influenza, or are these individuals more susceptible to, COVID, caused by SARS-CoV-2? Is there any difference that you're seeing there, or are we
Too
not there
early for
yet?
us to say. We can't make comparisons because we stay with one challenge model. I'm not focused on the microbes per se. I'm more focused on the immune response to said microbe. So we have done work with coronavirus. We have done work with other RNA viruses. We've done a lot of work with gram-negative and gram-positive bacteria. But I can't compare one versus the other and tell you one is worse.
No, no, that's all right. I just asked so many questions. It's just so interesting. So, what is, as an instructor, I know, teaching is a huge component
Mhm.
of, North Carolina Central's tradition, right? Of culture teaching. I mean, also research is huge, but teaching is a real huge component. What your students that you mentor, you advise, that you work with, what is their reaction to doing this type of work that is so closely connected with the community that they live in? What has been that experience?
Well, that I think is the reason why we're able to motivate students to work in my laboratory is what we do has a real world impact. And I think that that's a big deal. During the COVID outbreak and pandemic, we actually did a lot of community outreach here in the Durham area. And I went and actually I would give and educate about coronavirus and also the proper PPE necessary for protecting oneself. Did this at churches in the area, and my grad students did this as well. And I was able to get them out into the community, and it made them so much more impacted by what they were doing and the research they're doing. And I think community outreach and involvement is super important to, breaking down these ivory tower walls of what we do in a laboratory, what we do in a science environment versus what will actually improve the health of our community.
That's awesome. For the folks listening, just joining in, jumping in, Dr. Vijay Sivaraman at North Carolina Central University, going over his work about how environmental factors or behaviors influence respiratory health. It's just absolutely amazing. And you were talking about how you started to engage with these different communities. So that one part is, what are your students' reaction? The other part is, what was the community's reaction to you and your students that you advise, bringing this information to them? Are they aghast? Are their jaws dropped to the floor? Did they
Well,
realize that this was happening?
I think it's more comfortable. I think it's more comfortable. It's more real because it's not outsiders coming in and trying to educate. It's people from communities from the inside, like working with like, and that's I think an important factor. So
That is awesome. Now, so you have one arm where you're working with the community, but a lot of these things are kind of civil engineering kind of pieces, road construction, where are roads, the density of populations, our cars. You also talked about social economic pieces and how that then in turn impacts public health. With all the knowledge that you've accumulated and all the understanding that you will accumulate in the future, what are their plans, or what are your thoughts? How do you then generate change using the discoveries that you've made?
That's a good question. That is $1 million question. How do we help? How do we actually help our communities? And that is, it's a challenge. It's a struggle, right? It's a challenge and struggle because our communities have been developed a certain way because of a certain reason. And how do we repair that? How do we fix that? I mean, on one unfortunate situation is gentrification, right? Like our communities are becoming gentrified as property is so valued in the area. So the neighborhoods around NCCU are suddenly a focal point for communities that are of diverse backgrounds. And so you don't have it as a minority community. So now all of a sudden there's a lens placed on how to improve these problems. And with that comes redevelopment and potential reuse of redesign of roadway use. That's a negative way to look at it. What can we do in the here and now? Actually, I helped write a center grant that we submitted in January that we are going to continue to pursue as far as research focus, where we can actually evaluate the internal air, the indoor air of populations within our community, and help to regulate it. If we see that the PM levels are at a certain level or threshold, we're going to include like home air filters. And so if we can put air filters in people's homes, can we improve their outcomes? Can we bring down their particulate level exposure and then also improve their health outcomes? So we're actually going to use uh, real-time personal health monitors, smartwatches, as well as other kind of pulmonary expression systems to evaluate their outcomes as we have included the intervention. So we can see if there is a local environment that we can, local change we can make to improve people's lives. So that's something we'll be working on in the next year or so. So we'll see how it goes.
That is awesome, and I wish you all the luck in the world because I live in Durham, right? I know people from around the country will check this out, but I live in North Carolina. I live in Durham, and the work that you and your team are doing makes this community a better place for everybody to live in, but selfishly for me to live in. And no, honestly, I think when we think about research, when we think about innovation, one of the bottom lines is, wait, how does this impact myself and how does this impact my family? And just from me to you, thank you, Dr. Sivaraman, for the work that you do to make our community safer. And I know there's a long road ahead, and there are a lot of historical reasons why we are where we are. But folks like you are the reason why the future has an opportunity to be a better place. So thank you so much for that. But with that, I lead to my final question, and it's a curse of having great guests, is because the time just goes by like that. And I always end up kicking myself because there's always a question that I wish I would have asked that I didn't ask. And then I worry that my guest, they're leaving thinking, man, I wish he would have asked me this, or I wish he would have asked me that. And so I asked this one question at the very end of all of my interviews, and I'll ask this to you now. Was there a question that I should have asked, or a question that you hope that I would have asked that I didn't? I want to give you the opportunity to ask that question right now and then to just go ahead and answer it. So the floor is yours, Dr. Sivarana.
I'm sorry, sir. I don't have any questions to ask you. I am
No, you asked
honestly,
yourself. I'm the best podcast host. I asked you all the good questions.
well,
Like,
you did well. You did a good job of selling me, sir. I don't know. You made me sound really good.
no, you are
So,
really
I
great.
mean, look,
Go ahead.
what I really care about at the end of the day is I'm training students so that they can be the next wave of biomedical research scientists. I'm also training healthcare. I teach pre-nursing immunology and microbiology. I want to make sure that I'm training the next wave of healthcare workers. These are the bound, and especially in this economic and political climate, these people are going into battle without the information they need. So all I can do is prepare them as well as possible. And so that's what I hope to do. And along with that comes the research science, which will help to do kind of similar things, better inform our community as best we can. So,
Thank you so much. Last thing is, if folks listening wanted to do something to support research in general, support you, or to support North Carolina Central, what would you advise them to do?
well, on a local level, call your politician and tell them you care about science and you care about the status of our science and the funding of our science in this on a national level. And on a local level, give some money to the schools that are in your area. Help them out because they everyone needs funds to keep training students and to support students who can't support themselves. So
Well said. Dr. Sivaraman, thank you so very much for being with us today. Thank you for the work that you do in making our community safer. Thank you for the contributions to the science community in general. And thank you so very much for being with us on today's episode of the Richard Watkins Podcast. Again, I'm your host, Dr. Richard Watkins. And remember that the future is not something to wait for, it is something to build. Until next time, take care.